All right, hello everyone. All right, so I'm going to try to get the perfect distance away from this so my voice doesn't ring. Does that sound okay? Okay. As we too close, bad things happen. All right, so my name is Lightcap. I actually run a core facility at the University of Notre Dame. It is a materials characterization facility, and we are trying to bring more materials, more what we call beyond bio to the conference, and that has increased in recent years. And so it's been my job to also bring in those talks as well. So we had 3 really interesting talks lined up for today, and we have 2 speakers that made it. Our 3rd speaker had to cancel last minute, and that was Air Force Captain Rob Huthoefer. So he had made plans to join us today to share about the DURIP funding. I'm good? You want me to be closer? Okay. It's dangerous. All right. So there's a instrumentation funding opportunity from the DoD called DURIP, and he is a program officer within that program, and so he was going to tell us all the secrets to get new instrumentation. So last second, he was actually told, yeah, you can't travel anymore. So government efficiency stuff. So anyway, I think it's actually a mixed blessing because we have 2 other really interesting talks, and so they'll get a little bit more time. But also, I think you chose the right session too, because maybe we'll end a little bit early, we'll see, which is nice for dinner time. So before I tell you about the other speakers, I do have just some really short slides about DURIP, just so that you know a little bit about it if you haven't already applied or been familiar with this in the past. Okay, so here are the basics. One of the biggest misconceptions that I actually ran into with my own administration with this is they're like, well, if you're going to do a DURIP, you have to find a PI that has DoD funding already existing. Otherwise, they won't even consider it. So that's not true. Everyone I've talked to within the DURIP organization or within the DoD that has something to do with it said, yeah, that's not true. We do have priorities that we want to make sure that you're lined up with, and then beyond that, like if you have really good research merit and you have a great team and everything looks good, that's what we consider. So the biggest thing that I want to mention is that you don't have that requirement to have a PI with existing DoD funding. Like I mentioned, it's got to align with their current interests and priorities. And there's a solicitation like other funding opportunities that they put out there. And the awarded instrumentation is typically, it is better and it is more common for this instrumentation to go into shared facilities. So that's a plus for us as well. And it's administered by the Office of the Undersecretary of Defense for Research and Engineering. It's not a phrase I normally say. And it's in the branches for Army Research Office, the Navy and Air Force, the respective offices. Tips for getting started. So number 1, and this is really true with a lot of, if you've applied for like NIH or NSF grants before, start early. Contact with a program officer is really great because they can sometimes just like, that's probably not going to work. Then you can save a lot of your time. Or they might say, oh yeah, this is great. Let me give you some help, hopefully. So number 1, engage early with the program officers and then familiarize yourself with their priorities. Make sure that your research goals, what you can now do with the new instrument, what kind of research and new capabilities you can do, match up with the DoD's priorities. 2 that I mentioned just for here, there's a big biotechnical and medical research part, and also for us on the material side, there's material science and engineering, and there's some other things too that they're really into, like cybersecurity. And then you need a strong team of PIs, and then like anything else, just articulate research well, demonstrate that you actually have the need. Sometimes it's really good to put like, hey, here's what we can do right now with our current capabilities, and here is what we could do, send some samples to a vendor and be like, hey, what can you do for us with these samples, what kind of new results, new capabilities can you offer? And if you actually do a comparison in the paper where you say, like, this is where we're at right now, this is exactly what we'll have with the new instrument, and these are the things that will be enabled. That's really powerful. And then obviously demonstrate institutional commitment, and like any other proposal, cross your T's, dot your I's. I think that might be it. Yeah, that's it. Okay, so that was the first talk. I think maybe we got the record for fastest talk, even though it's not really for me, but yeah, go ahead.
Talk was way too short, so let's let them do it. So I've looked into this, and I should just say that my impression was that for life sciences, the Navy program is the most appropriate. Each of the different branches have their own application process. It seems life sciences is that, but for mass specs, for sequencers, for large-scale optical imaging instruments, this seems to be a really good opportunity. What I was wondering is if you knew when the application cycle was. I think it used to be at the beginning of September, but I thought it maybe moved to March, and I was wondering if you could clarify when the application cycle is.
I don't know. I did apply for one like 5 years ago, and I think it was in the summer or like late spring at that point, which we didn't get the award, but yeah, I don't know. And thanks for sharing. Very pertinent information. All right, cool. Let's move on. So our second speaker, and I think you're going. Yeah, right, because you're on this computer. Okay, our second speaker is Neila Gross. She is a PhD candidate and NSF graduate research fellow, so good job, in biomedical engineering at Boston University. Her current research takes aim at the mechanisms behind how microplastic contamination, which is found nearly everywhere in the environment, affects the rate of antimicrobial resistance. Some very scary stuff. Maybe we should dim the lights. Sorry. I'm also dad. Okay, sorry, I'm bad dad jokes. All right. So how microplastics affect the rate of antimicrobial resistance, and in particular, that impact on populations living in low-income and developing locations around the globe. As a part of that work, her research also examines best practices for the ethical frameworks guiding research conducted within vulnerable communities. And when she was an undergrad, she worked in my core facility too. She worked also as another research, like another research group, but she was very helpful like when we had a big external grant come in. And I think there was another session about how useful undergraduate students can be. So if you run a core facility and you do get external projects that come in, you probably have the experience like me that there's like a big flow and flux of demand. And staffing like in our core facilities is pretty constant. It's really hard to increase staffing quickly. Undergrads have been a fantastic way that I have been able to deal with large surges in demand. So typically I try to select a freshman or sophomore group of them and then train them, and then by the time they're seniors, I can pretty much give them anything. So a little bit on Neila too. So I will step down, and everybody please welcome Neila Gross.
Okay, now I'm worried about the mic. Okay, thank you, Ian, for that and your dad jokes. So my name is Neila. I'm currently a PhD student at Boston University in the Mohammed Zaman Lab, and today I will be presenting on some of the research that I've done regarding microplastics and antimicrobial resistance. Okay, so I know that not all of us are material scientists, not all of us are microbiologists or biologists at all, so I'm gonna go through a little bit of the background just so that everyone can be comfortable with the terminology and that we can follow along with the results together. So what is antimicrobial resistance? So in short, antimicrobials are basically any antibiotic that tries to prevent an infection. And so when you get a cut and that cut gets infected, you go to the doctor, and then the doctor will prescribe you an antibiotic. And so those work to basically halt the progression of bacterial reproduction, or even at times have cell death. So antimicrobial resistance occurs when these bacteria or whatever microorganism is causing the infection don't respond to the antimicrobial that you ingest. And so this can also take form in multi-drug resistance, or NDR. And so that is when the bacteria or microorganism doesn't respond to a variety of antimicrobials. And just to clarify, there are several classes of antimicrobials and several different types of families of antimicrobials. And so generally, if you have a resistant infection, you can go to a different family and it has a different target within the bacteria. And so you can generally pair together different antibiotics, and that gives you a solution to any antimicrobial resistance. And then to set the stage, the WHO, or the World Health Organization, announced antimicrobial resistance as one of our generation's most pressing health concerns. Okay, so what does this look like for us? Well, obviously, if the WHO declares it that much of a problem, then we know it's a very rapidly escalating health challenge. And we definitely see that it's unevenly distributed over the globe. So we see a lot of low-income countries affected more than their high-income neighbors. And so this was from 2019 as these stats, so they've increased dramatically since then. But in 2019, they found that you had around 8 million annual deaths due to infections. 5 million of those were indirectly due. So, right, you have an infection and, you have resistant bacteria, and so it's not treated, and then you get liver failure or kidney failure or whatever. And so that's an indirect death. And then around 1.3 million are directly due to the infection. So that's sepsis seated within your muscles, bones, whatnot, and you die of that infection. And so it's actually last resort antibiotics. There's 3 types. There's first line, second line, and last resort. So first line is whatever you're going to get anytime you get an infection generally, unless you already have a known resistant bacteria. And so the last line, or the last resort antibiotics, are antibiotics that doctors really don't want to prescribe, and they're very, very difficult to get, particularly in the United States. And so we want to try and keep those antibiotics isolated so that as bacteria get more and more resistant, we don't, they won't have seen the antibiotics, and so they'll be more effective per se. And so, well, resistance to last resort antibiotics are projected to double by 2029 now. So it's a sobering fact. So what can we do about this? So obviously, again, it's a huge problem. Most research right now is looking into creating novel antibiotics, novel treatments, not even antibiotics now. We're looking at bacteriophages, which are nature's response to bacteria, sort of in that check and balance system. However, the rate at which antimicrobial resistance is rising, our ability to create novel treatments is not keeping par with that rate. So that's a huge problem. So as scientists, particularly in my lab, what we're interested in doing is extending that runway, okay? So we have a runway of time where we get to the last antibiotic that we have that works. And so what can we do to sort of give other scientists who are looking into novel solutions more time? And so most research that does preventative work focuses mostly on human-human transmission, right? Like you get a cut, you do something, weird, like become blood brothers with someone else, and then they get infected. But at the end of the day, that's really not how most infections are actually transmits to each other, obviously. And so a lot of it is through the environment, right? So we, as I mentioned, you expose yourself to the environment, and that's how you get infected. And so our lab is particularly interested in the environmental dimensions of this. And one of the things that we're most interested in is wastewater, which, as you can imagine, has lots of bacteria. It's a great place to be at 8 in the morning sometimes. Anyway, so there are several environmental factors that are known to be perpetrators of antimicrobial resistance, but to just give you a few: heavy metals, biocides, which are like soap, antibiotic residues, obviously fecal contamination, and then biofilm formation. So the two that I'm going to focus on this talk are antibiotic residues and biofilm formation, but all of these are very, very important. Okay, so what is a biofilm? So biofilms are basically bacteria's ability to attach to a surface, and then they excrete what's called an extrapolymeric substance. So I kind of liken it to a house. Bacteria come in, and they hang out in this house that's very insulated and protects them from all types of weather or invaders, right? So in the bacteria's case, this is maybe mechanical disruption of a biofilm or an antibiotic itself, because that membrane is so tightly bound that you give an antibiotic as treatment to a biofilm, and it just simply won't permeate. So it is known that biofilms tolerate around 100 to 1,000-fold higher antimicrobial resistant concentrations of antibiotics. So the minute you get a biofilm, you're in trouble. But a good thing is that biofilms are really hard. It's really difficult for bacteria to form biofilms unless they have a surface to attach to. So in your bloodstream, that really hasn't been a problem because we don't have a lot of junk hanging out in our bodies, except that now we have a new player at hand, and that's microplastics. And so we all have heard, it's kind of a hot term right now. I feel like anything that has done anything with microplastics is immediately put on blast in the media, and there's a tablespoon in our brains and whatnot, which is very alarming, of course. But in this case, microplastics are a potential surface for bacteria to attach to and create this biofilm, whether that's in our bodies, outside of our bodies, and in our lab's case, we're more interested in outside of our bodies in wastewater. And so actually there have been some early field studies that have taken microplastics and sort of imaged them in a way where they've been able to see that bacteria are on the surface. Now, what type of bacteria, what they're doing is not fully known. The other thing that's not really known are the mechanisms by which potentially AMR that comes up via microplastics is done. So what we were interested in doing as a lab was first to sort of understand qualitatively what are the characteristics of microplastics that might be facilitating these high rates of antimicrobial resistance we're finding in the environment, and are they correlated or not? So in the study, we used 3 different microplastics. We used polystyrene, polyethylene, and polypropylene. Now, we know that plastics in general are almost impossible to get rid of, right? You can treat them with chemical, physical, biological, whatever, and they'll just disintegrate into smaller and smaller and smaller and smaller particles. And so we chose polystyrene because those are your packing peanuts, so it's kind of everywhere. It's also in like a lot of makeup and soaps. And polyethylene, which is your most ubiquitous plastics. That's like any of you having plastic bottles for your water or Ziploc bags or whatnot. And then we have polypropylene, which those are like carrying crates. And so these are the 3, probably one of the most common plastics found within the environment and also used by humans. And just a little bit about their chemical structure is that they're very good at stacking. Polystyrene is less good at stacking because of that benzene ring, but that makes them very, very, very resistant to any of the mechanical, chemical, or biological disruptions. One more thing to note is that polystyrene is the most hydrophilic out of all 3. So I'm sure you all know this, but like hydrophilic likes water, hydrophobic hates water. Plastics in general are very hydrophobic. That's the same for all of these 3. But of the 3 plastics, polystyrene is the most hydrophilic, which will come in later. Okay, so as I mentioned, we were really interested in looking at sort of the plastic characteristics, like the composition, concentration, and size, and how they affected the dynamics. And so, in short, we just varied all of these properties. And I do want to say, this was done in a controlled lab setting. This is not what's going on out in the environment. It might have a correlation to it, but in the lab, what we like to do is we like to control all of our variables before, so we can understand potential mechanisms or what's going on. So, how did we do this? Okay, this is really confusing, and when I did my PhD prospectus, this is what I got grilled on the most. It's just because it was difficult to understand. So I'm gonna walk us through this really carefully, and then hopefully that will help elucidate what's going on. So biofilms, right? Those are the guys that is like the armor, it's the house, we don't want them, but we're interested to see if they're on the microplastics. We used E. coli, which is a ubiquitous bacteria. It's everywhere, it's in your stomach, it's in the grass outside. Well, there's no grass in Las Vegas, but it's in the dirt. And so they're everywhere, and it's also a really great model organism. Everyone who does bacterial work likes E. coli because we know its whole genome, or most of it, it's annotated, and we know a lot about it. And so another thing that we know about E. coli is that they like to grow a biofilm in 48 hours. So what we did was we took this media, it's called LB, it's very like cell culture media, and we grew the E. coli with the microplastics in for 48 hours. We wanted to give it time to grow a biofilm and be able to, so that then when we started testing it, it would like start out with a biofilm if it grew one. And then after the 48 hours, we started what we called our antibiotic dosage, okay? So I mentioned earlier how you have low levels of antibiotic within the environment, right? You excrete antibiotic when you take them, right? Your body doesn't use all of it. But also, people love to not take the whole course of antibiotics. People love to toss it down the drain of their toilet or kitchen or whatnot. So they get out into the environment. So we have these really, really low levels of antibiotics, which allow the bacteria to sort of be introduced to them, but not enough to kill them. So I'm sure everyone here knows, like, you know, we gain tolerance to alcohol when we drink a lot of it, but not enough to kill us, right? And so your tolerance goes up the more you're introduced to alcohol because your body is evolving. Same thing goes, if anyone's seen The Hunger Games, President Snow likes to dose himself with a little bit of snake venom, so that if he's ever really poisoned, oh, I'm so sorry. I'm getting excited. If he's ever really poisoned, then he won't, die because his body has evolved to tolerate. It's seen the poison, it's seen the antibiotic or whatever, and so natural selection takes over, and the bacteria that are evolving around this tolerance are the ones that proliferate. So what we did was after that 48 hours, we introduced with the microplastics low levels of antibiotics. Okay, so we wanted to introduce the bacteria to the antibiotics, but we didn't quite want to kill them off. And then we tested their resistance levels using an MIC, a minimum inhibitory concentration. So the minimum inhibitory concentration, probably as you can imagine, tests what level of antibiotics will kill the bacteria. So as you can see, this is what this silly microplate dilution is. So in column 1, you're gonna have the highest amount of antibiotic. Column 2 is gonna be half of that. So let's say we start with 1 microgram per mil of ciprofloxacin. We'll have 0.5 microgram per mil in column 2, 0.25 and 3, 0.125, et cetera. I don't know what comes after 0.125. So we then put the bacteria into the plate and we see where they grow. What column are they gonna grow in? If they grow in, 4, then that tells me that that's the minimum amount, or the one, to the left of it, is the minimum amount of antibiotic I need to kill off the population, okay? Because they're not growing there, right? So we do this every day, and as we see an increase in this MIC, you can see that that would show that they're gaining resistance, right? They need more antibiotic to kill them off. And then the last 5 days, we took the microplastics and antibiotics out completely. And we wanted to see, okay, the MIC that they ended at day 10, whatever that was, whether they had resistance or not, do they keep that MIC without the stressors, right? Because if you evolve in any sort of way, if that evolutionary pressure is taken off, then generally your body won't need that gene or mutation or whatnot anymore, and so it'll get rid of it, because that's what's most, well, that's just how nature works most of the time. But if you do have that still, we call that a stable mutation, okay? And if it loses the resistance, it's an unstable mutation. Okay, so real quick, just to go into how we did the microplate dilution, it's cell culture. We're able to use a lot of our core facilities, like centrifuges and all of the different, they actually offer 96-well plates and pipettes whenever you need them. We do have our own, but it's really nice. So we take the microplastics, we vortex them down, and then we take the supernatant out, and those are all the bacteria, so those go up. Then we just have a tube just with the microplastics, and then we take the bacteria, and as I mentioned, we stick them into the microplate dilution. Each well gets about 10 microliters, so each well gets the same amount. Then we stick those in an incubator, read them with a plate reader the next day, and then that's how we tell who's grown and what's not grown. Then we do put a little bit of the bacteria back into the original tube, just so that it can grow up again. So that's kind of the gist of how that works. Now, what does the resistance look like? So this is just an example. So resistance is going to look like an increase in that MIC. So you can see the y-axis, we have a fold change in MIC. So instead of just giving the actual values of the antibiotic, we find it's better for like rate analysis and for a lot of the microbiology community to understand fold change. So you can see that the microplastics and LB, or bacteria, had a much higher and much faster rate of increase in their MIC, which means they needed a lot more, almost 100-fold more antibiotic to stop the growth of the population than when they needed at day 0. And then you can see that the ones without microplastics really didn't change a lot, which is to be expected. Right, and then we need to put that on a log scale because it's so drastic sometimes. So the next thing I want to show you, and I don't want you to freak out, but it's our multi-drug resistance results. And so we looked at 4 different antibiotics, ampicillin, ciprofloxacin, doxycycline, and streptomycin. Those are 4 different families, so they have 4 different targets of the bacteria to try and kill them. And then on top of that, we looked at microplastics and non-microplastics with or without the antibiotics. So I'll walk you through. So MICs are going to give us a really good idea of what's going on. I don't know why these graphs, the lines don't show up sometimes, but so what I want you to focus on is the black is the microplastics plus bacteria. The blue is just the bacteria, okay? And on the left, you can see that these were passaged in different low levels of antibiotics. So ampicillin, ciprofloxacin, doxycycline, and streptomycin. And then the MIC we tested them in is on the top going down. So you can see that top left graph is going to be bacteria that were grown with low levels of ampicillin tested in ampicillin. So the big takeaway from this is that the microplastics facilitated higher resistance than any of the non-microplastic samples, sometimes getting close to 1,000-fold, which you can see at the bottom right, with the bacteria grown in strep, tested in strep. But another interesting thing is that the bacteria that were grown in strep were also resistant to ampicillin and ciprofloxacin and doxycycline. And so these bacteria had never even seen these other antibiotics before, yet they were still very resistant to the antibiotics that they were tested in. So these would be classified as having multi-drug resistance, which is a huge problem. And so in the lab, we showed that it really, from the tested antibiotics that we looked at, it didn't really matter. The next thing that we looked at is the concentrations and size. So we looked at the different concentrations. We basically found that although they didn't have, if there were microplastics in there, there was no significant difference between any of the microplastic samples, but all were significantly different than the sample without microplastics at all. So what that means is basically from the concentrations we tested, which 1,000 microplastics per microliter is a field found value from different areas over the world. So that's a lot of microplastics. And so what we found basically from this is that it didn't really matter how many you had, they were still gonna get this insane amount of resistance compared to the ones that without the microplastics. Same thing for size. The sizes didn't really have a significant difference from each other, whether they were 500 micrometers, 10 micrometers, or 5, but they all definitely did differ significantly from the just the ciprofloxacin. And so, again, it doesn't matter what concentration, doesn't matter what size, that's a huge problem because you could then basically conjecture, right? If this is what's going on in the environment, you could have 10 microplastics or 1,000, it doesn't really matter. You're gonna get this really high number of antimicrobial resistance. The one other thing I want to point out is that 0.001, that dashed line there, that's the clinical breakpoint. So in clinic, when you are treated for an infection, we have set standards of when is an antibiotic no longer useful, right? You could pump someone full of ciprofloxacin, but they'd probably die if you got up high enough, right? It becomes toxic. So clinical breakpoint is when a doctor says, okay, we can't use this antibiotic anymore, we gotta go to another family, or we gotta go to a higher class. And so as you can see, all of the microplastics surpass that clinical breakpoint. Okay, the next thing we looked at was composition. How am I doing on time? Okay, so the next thing we looked at is microplastic composition. So we wanted to look at polystyrene, polyethylene, polypropylene. And we found that polystyrene had the highest number of resistance. And this was a really cool moment for me because I'd been looking at, well, I can't really see the microplastics unless they're the 500 micrometer, which is about 0.5 millimeter. And so I stuck the microplastics in our shared core facilities confocal, dyed them with a live dead stain, and I found a little world on the surface of these plastics, and even the glass to a certain extent. But the polystyrene definitely facilitated the most resistance by far, and it was significantly different from all the other plastics. Glass was really interesting too because it did give quite a bit of resistance. So we wanted to see, was this plastic specific or was it just a surface attachment thing? And what we found was that glass really didn't facilitate that much growth. The red indicates, so those are actually 2 glass microbeads right there. The red indicates a dead cell, whereas green indicates a live cell. So you can see that the plastics just facilitated a much, these were done at the same laser intensity, much higher, thicker, more live biofilm. So this was surprising because bacteria really like hydrophobic surfaces. So we thought polyethylene and polypropylene would dominate, but polystyrene is more hydrophilic. Turns out E. coli likes hydrophilic surfaces. But then we thought, well, wouldn't that make glass more of an issue? But it turns out it didn't. So there must be something really interesting about the plastics themselves that allow for like this facilitation of resistance. Okay, I'm gonna run through this because I do have some cool slides at the end. So why is this happening? What's the mechanism? Well, there's a lot that we could do. We could do genomics, we could do RNA sequencing, whole genome sequencing, we could do all sorts of things. But we wanted to look at the phenotypic change instead of the genotypic, or the genotypes changes. So We basically stuck the bacteria without microplastics, without antibiotics. We just wanted to see them grow. And we stained them with crystal violet, which basically shows you biomass, right? So something that's going to be more purple has more biomass, which means it has more biofilm. So you can see that the microplastic samples had way more biomass than the ones without microplastics. And that's shown with the plate reader we used in our awesome core facility. That you can see the microplastics have a much higher amount of biomass than those without the microplastics. So that showed that the bacteria were just better biofilm formers. And they were able to form a biofilm, and this is important, without a surface. So something, whether that was the microplastic or the environment that it was in, selected for these bacteria to be better biofilm formers. Without the microplastics even there. And this was a stable change. And so these guys, 5 days after, 10 days after, still had this ability to just coagulate around each other and form a huge biofilm and keep up the resistant levels that they were showing. So this is another, the confocal, it's a fluorescent confocal, so it can't show us biomass or of the biofilm, right? The green are the live cells, and so the EPS is full of lipids and proteins and carbohydrates and whatnot, so we can't actually see that. But we use an OCT in another core facility, and we get to see, I do want to point out, the glass biofilm, like it doesn't look like it's a lot, but that's actually a great biofilm. It's really like very thick. We really only want them to be 30 micrometers. These were grown for the same number of days. The ones with microplastics just grew this like insane biofilm so quickly, and it was sort of staggering to see almost. So, right, we would imagine that bigger biofilm, obviously more resistance. Those little red beads are 1 diameter polystyrene beads. And I know you're saying, well, why are those with the glass? But what we are able to do with both of those, so this was supposed to be a video, but it's not working. We can zoom in super close, around 60x on the confocal and take a video for 2 minutes. And then we plug that video into ImageJ, which can track the movement. And the movement is going to show us it's going to show us Brownian motion. And so, relative to the biofilm, of course. And so, when we track that Brownian motion, we can input that into a code that we have in MATLAB, and that will give us the Young's modulus of the biofilm itself. So not only are we trying to understand growth rates and AMR rates, but we're also trying to understand what are the mechanics of this in potential ways that we can start trying to view solutions for what's going on. And this all happens within the matter of hours. So this was a microplastic that we videoed for 30 hours, and the green are live cells that are actively attaching to the microplastic. So something is going on where it just provides an environment that's really amenable to the bacteria to attach to. And this was also done with the confocal. We're awful. We're on their total bad, naughty list because we use it for so long, and it's a shared facility, and it's really bad of us, but yeah. Right, so from just basically the gist of this is from these studies, we were able to see that microplastics facilitated clinically relevant multi-drug resistance via biofilm formation. And then the relative size and concentrations we tested really didn't matter. Polystyrene was by far the biggest facilitator of antimicrobial resistance. And then bacteria have a phenotypic change that make them better biofilm formers. So I will actually have to sprint through these slides, but really quickly, what we're gonna do next in the lab is we have a lot of partners around the world. We're currently based in Tirana in Albania near a forcibly displaced camp. Obviously, trash, waste disposal, any sort of infrastructure with vulnerable communities is just simply not going to be present. And so these are the environments that are going to facilitate the highest amount of antimicrobial resistance. And at the end of the day, this affects us all. And so We have places in Pakistan, in Karachi, in South Africa, in Durban, in Kenya, in Kakuma refugee camp, as well as a couple other places around the world where we have partnerships with local teams to try and do surveillance and whatnot. This is one in South Africa. This is just how we'll do it. And yeah, we want to continue doing more research, understanding the material properties of the microplastics. BU, again, has a Raman spectrometer and a SEM that will allow us to look at more of the material side of what's going on with the microplastics themselves. We want to do lots of different sequencing, and BU has a great core facility for that as well. And then we'll do more confocal things because it's pretty. Oh, so that's an SEM of a microplastic, and then, sorry, at the top, that's the Raman's, it's a Raman's fingerprint of a microplastic, polystyrene microplastic. Okay, cool. So basically, this whole talk was for me to rant about my research, but also to just show you how like multifaceted different projects can be, and how important it is that we have core facilities that support little undergraduate Neila and grad school Neila. And so, yeah, so I do want to thank BU's core facilities, the mini core and the MPC, which are awesome.
Let's thank Neila. So while the next talk is getting set up, Neila, I don't want to talk to Don. I thought you would have a chance for, like, while we're doing that, just like a question or two, if you have a question.
Great talk. My name is George Bill. I'm at the University of Miami. We've been doing, I thought I had my voice loud enough. Okay, so we've been doing a lot of wastewater sampling since COVID, and we have a lot of targets besides SARS-CoV-2 now. One of the projects we've done is for hospital wastewater, correlating AMRs with the amount of antibiotic prescriptions in the hospital, and there is a very nice correlation. And we've stored all this water. We have a biorepository of all of this. One thing that we didn't look at was microplastics. That sounds like a good thing to do retrospectively, and I'm happy to talk to you offline more about possible collaboration in that area.
Yeah, there's all, there's so many things, but yeah, it's like low levels of antibiotics plus bacteria is like the worst thing ever. So yeah, that'd be great.
I'm wondering if you have any theories about what actual phenotypic change is happening here to create this kind of functional adaptation.
We did do some genetic testing, and we did have some mutations in the motility function, so their motility was just a lot smaller because once you're in a biofilm, you actually don't need to move around as much, so they lose that function, and then what they gain is in that are better bioconformers. And so, I mean, that can also just be awful for a variety of reasons other than being a bioconformer, but yeah, it is. There's like, it's such a new field that it's just like, wow, everyone has to go through. So, yeah.
Thanks.
Let's take one more time. If you guys do have questions, like point out. Okay, I'm going to introduce our next speaker. Looks like we're ready to go. So Professor Tom Zega is a faculty member with a dual appointment in the Department of Material Science and Engineering and at the Lunar and Planetary Laboratory at the University of Arizona. His research employs microanalytical techniques to unravel the mysteries behind the chemical and physical evolution of the early solar system. As a part of that work, he has analyzed samples from the JAXA Huthoefer missions to asteroids Zega and Zega. And today, he's going to share the story of NASA's OSIRIS-REx mission to retrieve samples from the asteroid family. He will also share about his role as played in the analysis of those samples. So please welcome.
Okay, thanks Ian, and thanks Neila for a great talk. Just disclaimer, I am not a biologist. I'm a lowly geochemist and material scientist, so I'll do my best to entertain you. And Ian, you'll have to keep me on time. I'm fully aware that I stand between you and happy hour or dinner or whatever you fancy this evening. Let me just give you a brief introduction about OSIRIS-REx. It's an acronym, and it stands for Origin, Spectral Interpretation, Resource Identification, Security, Regolith Explorer. And these all fold out with different objectives. So for example, Origins, the goal was to return and analyze a sample of pristine carbonaceous asteroid regolith. Regolith is just a word for rock blanket. The other goal for spectral interpretation was to provide ground truth for astronomical and telescopic observations of the entire asteroid population, and I'll tell you about asteroids in just a moment. We also aim to map the chemistry and mineralogy of a primitive carbonaceous asteroid to see if there were any resources that might be identified in situ. We also wanted to measure something called the Yarkovsky effect on a potentially hazardous asteroid. The Yarkovsky effect is just this basic process where asteroids will radiate thermal heat on a diurnal cycle, which will change their orbital characteristics and potentially make them Earth crossers. So we wanted to measure that in situ. And then we also wanted to image and map the chemistry and the boulder sizes and granularity of the asteroid down to the sub-centimeter scale using remote sensing cameras. And OSIRIS-REx was a $1 billion mission. It was selected in 2011 as part of NASA's New Frontiers program. So this is the timeline for the OSIRIS-REx mission. So we launched in 2016. This is a picture of my son when he was about 10 years old. He's actually pointing to the launch tower right there. And we were there that day. It was a marvelous day at the Kennedy Space Center. And there have been a few times in my career where I've been truly inspired by some of the science that we get to do. This was definitely among them. So we had a 2-year outbound cruise to the asteroid, and we entered into orbital insertion sometime around late 2018, 2019. We were there for 2 years, imaging the sample surface at high resolution, doing infrared spectroscopy, measuring its chemical makeup, and deciding exactly where to land on the asteroid and sample a piece of it. This all happened over the pandemic, which now when I reflect on it, and probably many of you do, was a very strange time in the history of the world. And it was even made stranger trying to do an asteroid sample return mission all remotely over Zoom, WebEx, Teams, whatever your favorite remote meeting application is. We departed in early 2021, and we returned over 2 years to Earth, and sample return was in late 2023, September. And then this is a picture I took from the observation site. You can see the rocket starting to turn over as it goes into low Earth orbit. So if you don't know anything about asteroids, that's quite okay. We can orient ourselves here. So this is a sort of top-down picture of our solar system. Of course, the sun is at the center, Mercury, Venus, and Earth, the third rock from the sun. Then there's Mars. And between the orbit of Mars and Jupiter, which is somewhere out here, is this large belt of asteroids, the main belt, as we call it. There are over 1 million asteroids that we know of right now in the main belt. There's millions of unknown. The largest of these is Ceres. We visited Ceres, and then the next largest is Vesta. Ceres is about 1,000 kilometers across. Vesta is about 500 kilometers across. And these asteroids are constantly being observed, and new ones are being identified all the time. The total mass of the asteroid belt is something about 4% of the moon's mass, to give you some perspective. Okay, but then there's also near-Earth objects, or NEOs, or NEOs as they're sometimes called. There's about 40,000 of them as of today, and greater than 90% of NEOs are actually near-Earth asteroids. So all these green dots here are asteroids that are near Earth, and then the red ones are actually hazardous asteroids that we want to keep an eye on. The rest of these are near-Earth comets, and comets have very strange elliptical sort of trajectories, which are indicated by these various ellipses for different comets. So OSIRIS-REx went to asteroid Bennu. Bennu is a NEO, it's a near-Earth object. At the time of its selection in 2005, there were over 500,000 asteroids known. As I just said, as of today, there's something like 1.5 million. But at the time, there were 500,000 asteroids. Over 7,000 of those were near-Earth asteroids. About 192 of them had optimal orbits for sample return. And then about 26 of them had a diameter greater than 200 meters. Why that matters is because those smaller than about 200 meters often have very strange rotation rates and orbits, and they're not very safe for us to land on. And then 5 of those were carbon-rich, and we were interested in carbon-rich asteroids because they would contain organic compounds, and they may tell us something about the origin of life. And so Bennu itself rose to the top based on ground and space-based astronomical characterization. And there were some key questions we wanted to answer. So for example, what role did asteroids play in the origin of life as we know it? And perhaps how did they serve as fundamental building blocks for planet formation? Yeah, it's just simply because the observational campaigns that have occurred over the past 15 years, they keep identifying increasingly smaller asteroids that add to the total population. Absolutely. Yeah, we just didn't know about them yet. And if you've ever seen any of the Hollywood movies like Deep Impact, or I don't know what the other ones are called, they sort of embellish the hazards of these things, but it is important for us to know what their characteristics are in case any of them are Earth crossing and we can mitigate those hazards. Okay, so this is what asteroid Bennu actually looks like. It's a carbonaceous, what we call rubble pile asteroid. It has a spinning top shape, so if any of you remember as children, you played with the tops, spinning them, hence the name spinning top. As I said, it's a near-Earth object. It has a very rough, sort of boulder-dominated terrain to it. And we spent about 2 years surveying sites on the asteroid. We identified 4 potential ones, and the one we chose to actually sample was nicknamed Nightingale. And then we acquired sample on October 20th, 2020. So to give you some context, asteroid Bennu is about 500 meters in diameter relative to some obvious landmarks like the Eiffel Tower in Paris or the Empire State Building. It's actually quite large. Correct, Ian. And sample return was the primary objective of the OSIRIS-REx mission. The touch-and-go sample acquisition mechanism, or TAG-SAM, was the device that we used to actually touch or tag the asteroid. If any of you are gearheads, like I sort of am, it might remind you of old-style carburetors from cars during the 60s, 70s, 80s, and maybe even into the 90s. So basically the idea is that this TAG-SAM would touch down on the asteroid, nitrogen would flow through the center of this annulus here, it would fluidize the surface, the regolith with nitrogen, and then particles, and this is just a test device here, particles would become entrained within this TAG-SAM. And the key requirements for the mission were to return greater than or equal to 60 grams of pristine bulk sample from the surface and or contact greater than 26 centimeters squared of the surface and return it to Earth. And I'll show you some pictures of that in just a moment. From the orbital phase, using remote sensing cameras and infrared spectroscopy, so this is near infrared on the x-axis and wavelength micrometers, and then reflectance on the y-axis, we knew that Bennu itself probably had hydrated material on it, so the red spectrum here is Bennu, and then these other spectra, green, orange and red, are from analog meteorites that we've measured in the lab, we have here on Earth, and we can look at in more detail, and we know that they have a major water absorption band at somewhere around 2.5 microns. We also knew from orbital phase mapping that there were abundant oxide materials, so this is a spectrum of Bennu in black here. This is a spectrum from magnetite. Magnetite is just Fe3O4, and you can see some of the similarities in absorption between Bennu and magnetite, suggesting that magnetite was present on the surface of Bennu. And then we also looked for organic compounds, so this is from 3 to 3.5 microns, again reflectance, and Bennu's in red here, and then these black curves are with reference to some organic compounds, and apparently there's absorption bands for different CH bonds and stretch features in the infrared. We also identified carbonate on the surface, so this is a picture of Bennu's surface, and some of these bright, what were called specular features, were mapped again in the near infrared. So Bennu appears in black in each one of these spectra, and then the colored spectra here are for different types of carbonates. So calcite is calcium carbonate, siderite is iron carbonate, brunerite is an iron manganese carbonate, dolomite is a calcium magnesium carbonate, and then magnesite is a magnesium carbonate. And so you can see the similarity of some of these reference materials to Bennu spectra, suggesting that maybe some of these features were in fact large-scale carbonates on the surface of Bennu. We also mapped the surface and we looked for carbon-bearing materials. So some of these maps here show the distribution of carbon. These are sort of false color where enhanced red values indicate enhanced amounts of carbon-bearing material. And then blue indicates hydrated sheet silicates. So these are silicates that actually have structural water to them or hydroxyl groups in their structure intermixed with organic compounds on the surface. So the touch and go happened on October 20th, 2020, and I have a video here. Hopefully it works. I think it will. So this is actually a reproduction of what happened here. All these features here that you see were boulders that we had to navigate around. There's the tag SAM. It touches down, it fluidizes the regolith with nitrogen gas. There's this explosion of particles. Materials collected into the tag SAM, and then 9 seconds after contact, we fire thrusters, we halt the descent, and we start backing away from this explosion of particles. The arm itself sank 1/2 meter into the surface. We didn't realize that was going to happen. It was far deeper than we expected, suggesting that the surface was incredibly weak. And then 16 seconds after that, we fully emerged from the surface and we started to back away with this hopefully handful of material after kicking up about 6 tons of loose rock. And then about 30 seconds afterwards, we shut off thrusters and we drifted away back from the surface and then started thinking about our return to Earth in 2023. Yeah, NASA's pretty good at this sort of stuff. And it's really effective for things like this, right? So this is the Nightingale site before we touched down, and then this is what it looked like afterwards. So you can see we obviously perturbed the surface pretty well. I can go back and forth several times. So pre-tag in March 2019, after tag in April 2021. So this was after plunging about 500 meters into the surface, and the engineers and flight crew with Lockheed Martin who controlled the spacecraft suggested that if we hadn't fired back-away thrusters, we might have gone all the way through the asteroid. It's that loosely consolidated, which we were not expecting before we tagged the surface of the asteroid. And then when we backed away from Bennu, we were, with cameras on the spacecraft, we were actually able to image the bottom of the tag SAM. You'll notice these round things on the outside. Those are called contact pads. They're stainless steel Velcro. And the idea was that the surface particles would become entrained in that stainless steel Velcro for looking at surface processes like space weathering. But what we noticed was that there was a rock or stone that was blocking one of the flaps from closing, and we were losing particles before we stowed the tag SAM inside of the spacecraft. Because it's so fluid under these microgravity conditions, particles just wanted to escape out of it. And so we thought, we better close this thing, seal it up very quickly before we lose any more mass. And the estimated mass loss from this was about 10 grams. So this is what it looked like when we got it in the spacecraft. We closed the heat shield on top of it. The estimated sample mass that we returned was about 250 before we got to Earth, but the actual return, once we measured it at Johnson Space Center and NASA, was 121.6 grams. So to put that in perspective, that's about enough to fill an 8-ounce coffee mug, which you might say is not a lot, but the science we get from that is absolutely astonishing and rich. Okay, and we expected some basic mineralogy and other types of things from that, which I can show you some pictures of. Sample return happened in 2023 in September. So the landing site was in the Utah Test and Training Range, which is just outside of Salt Lake City, Utah. So we had a landing ellipse, and then we had a processing hangar for temporary storage before transporting the spacecraft to NASA Johnson Space Center in Houston, Texas. Some components of the spacecraft went back to Lockheed Martin for examination and archive, but the sample itself went to JSC. Here's what it looked like arrival day. This is Monday, September 24th. So I was not part of the recovery team. I was part of the initial analysis team. And so when the spacecraft was entering Earth's atmosphere, I was literally at home in my living room waiting to drive to the airport to get on a plane. So I was watching this happen on NASA TV, but I was there on Monday morning or afternoon when the spacecraft finally arrived in the truck. They started transporting it into Building 31, which is where the curation facilities and some of the analytical facilities are located. So it was opened on September 26th, which was Tuesday morning. This is what the tag SAM looked like with the avionics deck inside the glove box. This is a nitrogen-filled glove box at Johnson Space Center. And we found that the outside of the touch-and-go sample acquisition mechanism and the tag and the avionics deck were actually coated in this black fine grain dust. And the sample analysis plan that we had developed over several years hypothesized that the dust was broadly representative of the bulk sample. So maybe this is a better picture. You can see some of this black dust here. So it's all sort of coating the outside of the tag SAM itself. And so we got to work, the curators collected some of this and we got to work on that shortly after its arrival. And so there was this quick look team that we assembled to do measurements, and the goals were to inform the public, perform preliminary investigation for science purposes, and guide ultimately sample selection. So just some pictures. So these are some of the shared facilities at NASA Johnson Space Center. So believe it or not, we still do visible light microscopy. We do scanning electron microscopy. We do Fourier transform infrared spectroscopy, X-ray computed tomography, UV fluorescence spectroscopy, X-ray diffraction, and a few other types of measurements were done for this quick look. I won't get into the details of all the samples, but there were a number of different types of measurements that were made. This is what some of the allocations look like, and you can make out that the sample from Bennu looks like this sort of fine grain, very, very black, very dark, dusty material. X-ray diffraction revealed a number of different phases. So this is degrees to theta, so 5. I think we scanned up to about 90, but only up to 75 is shown here because that's where the principal diffraction occurs, and intensities on the y-axis. The major phases are hydrated silicates, iron sulfides, magnetites, carbonates, and anhydrous silicates like olivine. The sheet silicates, which contain water, or a mixture of different types of silicates. There were some phases we expected but were not detected automatically. But this showed that Bennu had a diversity of material and in fact was incredibly hydrated. So just some pictures of what the fine grain fraction looked like. It's dark, it's got small grain sizes, and it has some highly sort of reflective phases as well that turned out to be iron sulfides. There was also a coarser grain fraction that we looked at, and one of the more interesting particles that showed up was some of this bright isolated stuff that you see here, which we initially thought was contamination. And contamination control was a real significant concern on the mission because we wanted to make sure what we were looking at was in fact native to the asteroid and not some contamination from the spacecraft. And it turned out that these things were magnesium phosphates, which was a complete shocker, and to this day remains a very key focus of sample characterization for the mission. So we did some SEM. We prepared samples in different ways on beryllium planchettes, low background holders, some that were more conductive like carbon, and we looked at the particle makeup. So this is just some SEM imaging and EDS analysis of particles. They all have obviously very rough surfaces, so the X-ray spectroscopy that we did was not ideal because these were not polished smooth surfaces, but we can make out the basic sort of phase distribution and mineralogy of the surface. So for example, you see lots of magnesium silicates, you see spatially correlated iron and sulfur suggesting sulfides, and then calcium, which would track to things like calcium carbonates. When we look at it in the SEM, there's a really fine-grained matrix to it. There are suggestions that there's small variations in carbon abundance in the matrix, which probably trace to organic compounds and which since have been characterized and recently just published on. There's these interesting wispy textures in the fine-grained fraction, suggestive of sheet silicates and hydrated silicates. FTIR also confirms that we have hydroxyl groups in some of these structures. There's also weak aliphatic bands present, suggesting chain-like organic compounds in the sample. There's some really interesting iron oxides. So these are interesting morphologies, plaquettes, these sort of dodecahedral structures of magnetites. There's framboidal magnetites as well, but all are pretty pure Fe3O4. There's lots of iron sulfides in the sample. So we find these hexagonal pyritites, which is Fe7S8, some more platy and have twin boundaries all over them, which is really interesting to find. Some more iron sulfides. Some of them occur as veins in the sample, suggesting fluid flow in the asteroid at some point in its history. And then calcium carbonates, like I said, as well throughout the sample. Okay, so there's a lot of words here. It's kind of a word salad, but basically all this says is that the minerals that we find in the sample are very characteristic of hydrothermal processes. We know that phases like sheet silicates, phyllosilicates, carbonates, magnetite, pyrrhotite, all of these things form on Earth through aqueous phase processes, okay? This suggests that at some point in the history of this asteroid, it had a very active hydrothermal system, and the asteroid itself bears a startling resemblance or similarity to some meteorites that we have collected on Earth that also suggest previous hydration and hydrothermal systems. So that was the story as of June 24. Since that time, NASA Johnson Space Center has allocated samples to the science team, which is spread across the globe. You might ask what we've found since then. We find things like salt minerals. So I've mentioned things like carbonates and sodium phosphates in the sample. So these are just some examples of the things that we find. We find sulfates, we find chlorides, we find halite, which is table salt effectively, sodium chloride. We find fluorides, and all of these form during evaporation of what we call late-stage brines that probably existed early in the history of this particular asteroid. And these are some other examples of sodium carbonates and calcium carbonates. Some of these have really interesting morphologies to them, like veins, sheets, some needles. And we came up with a model of basically how we think this evaporation process could have occurred on the asteroid. And we hypothesized, in fact, that maybe similar brines still in fact exist on the surface or the interiors of icy bodies like Ceres, which is a large asteroid, or Enceladus, which is a moon of Saturn, which is indicated by spectra or measurement of their icy plumes as we see them. So to sort of bring this whole thing home, shared facilities obviously have played a significant role in the characterization of the sample. So our particular facility at the University of Arizona is called the Kuiper Arizona Laboratory for Astromaterials Analysis, or K-alpha for short. And so any of you that have done electron microscopy will obviously recognize the play on words here. So we have capabilities from the sort of visible light scale all the way down to atomic, including electron microprobe, scanning electron microscopy. We have Raman with SEM. We have a nanoscale secondary ion mass spectrometer for trace element analysis and for isotopic analysis. We have an aberration-corrected STEM for atomic scale characterization. And we can handle a variety of particle or sample geometries for sample characterization. For any type of planetary materials or any material in general, because we have to do this sort of coordinated or correlative microscopy to extract the maximum amount of information from these samples, which are obviously very, very precious. And building the facility took many years, many proposals, and a lot of frustration, as probably all of you can relate to. The overall management is by a scientific and technical oversight committee. Yours truly is the director. The instrumentation was funded by NASA and NSF. Our annual operations are somewhere around $1.5 million. We have 4 full-time professional staff. We have a business manager and IT manager. And we're financially supported by the University of Arizona's Lunar and Planetary Laboratory, the College of Science, the Office of the Vice President for Research, and NASA has a program now called Planetary Science Enabling Facilities. So we get the bulk of our operation expenses from that. And then, of course, we have an hourly recharge as well. We support materials characterization across campus with the heaviest use from obviously science and engineering. And researchers with a NASA grant or contract can use instrumentation free of charge. Those without a NASA grant or contract pay a nominal hourly fee, which is subsidized by the university. And obviously, the facility has been critical to characterization of all kinds of materials, including those returned from the OSIRIS-REx mission. And we're still ongoing. We have a bunch of driving hypotheses for the mission. I'm not going to read any of these, but they all relate to the asteroid's history. But I think just to sort of bring this home, there's an interesting video that ULA put together about the mission itself. And it just shows the sort of timeline for the spacecraft and the rocket. I think it shows the launch eventually. So here's the rocket being towed out to the launch tower. A lot of concerned engineers. So this was a few weeks before we launched. None of the science team was allowed out here except for the principal investigator. So eventually it gets to the launch tower. ULA was the contractor for the rocket. I think the rocket costs like $250 million if anybody's interested. And then they have their launch sequence.
Status check to proceed with terminal count.
So this was at Cocoa Beach in Florida.
Go. Hydraulics. Go. Pneumatics. Go. LO2.
Go.
Vehicle system engineer.
Go.
Anomaly chief.
AC is go.
Range coordinator.
Clear to proceed.
Launch director.
LD is go, and you have permission to launch.
Proceeding with the count. Status check.
Go Atlas. Go Centaur.
Go OSIRIS-REx.
5, 4, 3, 2, 1. And liftoff of OSIRIS-REx, its 7-year mission to boldly go to the asteroid Bennu and back.
It was a beautiful day that afternoon. The flight had been scrubbed the day before, but that day turned out to be perfect weather in Cocoa Beach, Florida.
Reach and pressures have plateaued. We've done around. The program is complete.
And you see the solid rocket booster jettison.
Separation
So
was
now it's in low Earth orbit, or just about there.
and full thrust on the aero fin. And we have indication of spacecraft separation.
And there it goes. So just to conclude, OSIRIS-REx has been a 14-year-long mission. In my opinion, it represents the height of human imagination, creativity, and ingenuity. It's remarkable when a team of people can get together and plan something like this and be supported. It's a remarkable success story. It's not over. We still have a lot to learn to tell the story on our origins and rewrite the textbooks. And shared facilities have obviously been very critical to its success. So if you're interested, stay tuned. And with that, I'll thank the organizers of the session for inviting me to come and speak, the OSIRIS-REx science and engineering teams, various colleagues and scientists on the science team, NASA, NSF, the University of Arizona, and then Lockheed Martin for actually building and operating the spacecraft. And thank you all for your attention.
Because our session is over, I'm just going to say we're not going to take questions, but if you have questions, feel free. I don't know if you can say.
Yeah, I can.